So you have heard science talk and technology talk. Of course, EMSL is a user facility. So user is very much at our heart. So we collaborate with users, we help them, we advance science. The next speaker will be one of the users. Himadri Pakrasi is a distinguished university professor and director of International Center for Energy, Environment, and Sustainability at Washington University in St. Louis. Himadri has been highly productive, a high-impact EMSL user, and he led one of the EMSL grant challenge John mentioned earlier, and he focused on Cyanobacteria, Cyanobacteria use in solar energy and drive the chemistry of life. So Himadri will tell us about the results of that grant challenge. Himadri.
It's a distinct pleasure to be here. My thanks to the current EMSL director, and Dawn Baer has done such a great job in organizing this, this meeting. Everything is just perfect, and I know that Dawn and his team spent a lot of time on getting this thing going. I am not very good in talking about history because I very seldom do that, but this gives me an opportunity to think back a little bit. I'm not going to talk about a specific time. I'm just going to go by millennium because we started our project right at the turn of the millennium, at least the ideas around that. So it's the past millennium and this millennium. Let's just go with that. So around the time, BER and other agencies were thinking about building large facilities. Some of us remember those endeavors. And at that time, I got attracted to that because biology was just entering what I will say the big data phase. Lots of data that were being generated or about to be generated. And the question was one of how can all of those data be used in an effective way to understand biology in a very fundamental way. So I remember in those days, I had the good fortune of talking to many individuals who are thinking about that, and Steve Wiley is sitting here. I remember at some of those meetings we were discussing all of this. But typically I have been rather impatient. I didn't want to wait for those facilities to be built. And I learned about EMSL that here is a place where there is already a facility where there are lots of instruments that can be used and can be used collectively to devote at least some part of these different instrument times to solve big biological questions. And so this was the impetus. I remember coming here and good to see Bill Rogers sitting here, and I walked into his office and was introduced to the EMSL concept, turn of the millennium again. Allison was still not the director. And then there was an ad that came out in Science where there was a call for proposals. And in the meanwhile, I have been talking to the friends and collaborators that I have known for quite some time and said, let's give it a go and put together a proposal. Our greatest fortune that ours was selected at the outside Grand Challenge project, and that was around membrane biology, and it was called the Membrane Biology Grand Challenge. So that's kind of the history. So here are the cyanobacteria that I'll be talking about. And when we started, this was the statement that we made. It's a partnership between a DOE National Lab user facility and university research groups in a systems microbiology project. And I was fortunate to be the lead person for this entire project. And I did not know really well how national labs really work. So at that time, came over here and met Dave Copinol, and we became the co-leads. And all I can tell you is that, even after all these years, Dave and I, we try to get in touch with each other, if not every week, at least every month. And we get in touch with each other to conduct more exciting science. So that was the beginning of that. This is a slide that many of you may remember. This was given to me for my first presentation on behalf of MBGC. And this picture has changed, I'm sure. These are probably ancient instruments at EMSL. But I keep this picture in my mind because it's this business of unique tools and unique science. When I first came here and I was given the opportunity to talk to all the instrument, the capability leads, I was literally a kid in the candy store. I did not know what I should not be using. And so basically it's all these tools, and the challenge was, how can all of these tools be applied in an effective way and collaborative way to answer the challenges that we had in this area of cyanobacterial membrane biology. Here was the team that we had. We had Washington University in St. Louis PIs, Lou Sherman, who is sitting here from Purdue, Tom Smith from St. Louis at the Plant Science Center, and Reggie Aurora from St. Louis. And we also had Teruo Ogawa, at that time associated with an institution in Shanghai. We were the outside group of PIs. And then from EMSL and PNNL, there were a lot of individuals. But I want to point out some of the names here, and maybe some of these individuals are sitting here. I know some are. Dick Smith is one person who influenced me a lot in the earlier days. We used to have conversations about the proteomic capabilities, but this was a team effort. And I want to acknowledge the fact that our project, the way it developed, it was all because of somewhere around 50 or 60 individuals from different academic institutions and here at EMSL and PNNL coming together and attacking this big challenge. I will give you just a little bit of information about all that happened. But before I forget, let me just tell you that in those 5 years, we ended up publishing together more than 50 peer-reviewed journal articles. And the more important thing is that those articles were not just a single authored or single PI article. These were truly representative of collaborative efforts that were led, that were done in the context of MBGC. Okay, so let me go in and talk to you about the system that we use. We used a photosynthetic microbe, and here are the different kinds of photosynthetic microbes that are shown: an oxygenic among those cyanobacteria that we'll talk about more. Then also eukaryotic algae, they are also microbes that are doing photosynthesis. Now photosynthesis, especially the oxygenic photosynthesis, it evolved only once. The basic machinery of photosynthetic, oxygen-evolving photosynthesis evolved once, and evolved in cyanobacteria that used to live at that time, it's believed, in water. Cyanobacteria today lives everywhere, on soil, in water, at very high temperature, at very low temperature. And that's simply because cyanobacteria are the ones who figured out there is this abundant source of electron, and that's called water. This was the organism that invented the whole business of splitting water to get the electron, using the energy from light, and then make good stuff that you and I, all of us, are dependent on. And there was a byproduct called oxygen. So this very simple reaction is what has intrigued me throughout my scientific career. And we decided to go after this, but asking a question about cyanobacterial biology that was intriguing at that time. So let me introduce you to that one. So again, cyanobacteria are oxygenic photosynthetic prokaryotes. They are known to be the progenitors of chloroplasts in plants. Abundant fossils in Precambrian rocks. They have been around for a very long time. And they have been found in most diverse ecological niches because of what I just told you, because all you need is just plenty of sunshine, carbon dioxide, which we have plenty of, and probably are going to have more of, and then just water. So they can grow everywhere, low and high temperature, on very dry to very wet conditions. And the important thing that was discovered toward the end of the last millennium was that these were the only known bacteria with circadian lifestyle, just like you and me. So they undergo diurnal rhythms just the same way as we do, the plants do, and many other eukaryotes do. To date, these are the only prokaryote or bacterial systems that are known that have circadian lifestyles. So we thought that we will get into this and try to understand how cyanobacteria, what are the determinants that allow them to conduct the circadian lifestyle. Again, this is something that I think most people in this audience know about, but I think that important thing for us to again remember that cyanobacteria have been responsible for the oxygen-rich environment that we have on this earth. These have had lots of ramifications in the Earth's history. Let's go to the cyanobacteria that we have been studying. Okay, so there are different types of cyanobacteria. This rectangle is supposed to be a micron. So these are 4 micron or 2 micron or 1.5 micron or maybe as large as 10 microns. Who are they? This is an organism that I'm just going to briefly mention. This is a workhorse among the cyanobacterial model systems called Synechocystis. I'll come back to this later on. The one that we chose is called Cyanothece 51142. This was isolated in Lou Sherman's lab in the early 90s, and then the pioneering work was done in his lab at the time when MBGC started. We knew that it has diurnal rhythm, but we did not know what are the determinants and especially what were the networks that were governing these diurnal features. Another thing that was very important about Cyanothece is that they can fix nitrogen. All cyanobacteria produce oxygen, and some cyanobacteria can also fix nitrogen. Cyanothece 51142 was one of them. So unicellular cyanobacterium, and that's important, capable of oxygenic photosynthesis and nitrogen fixation. And this was a big question at that time. Photosynthesis produces oxygen, but nitrogenase is oxygen sensitive. And we are talking about a single cell organism. So how is it that this single cell organism manages to do both of these? And the answer, here's the answer. It's the robust diurnal rhythm, photosynthesis at daytime and nitrogen fixation at night. And you can see already how these bacteria are using very clever way their circadian lifestyle in achieving these two incompatible but very important roles, fixing carbon and fixing nitrogen. So we started looking at the inside of the cell. By the way, they can grow very well, even better in reactors. Here is a TEM, basically showing that inside the cyanobacteria, there are lots of membranes, and I will show you a better picture of this. But it also has all of these internal storage bodies, glycogens, polyphosphate, and then also cyanophycin that I will talk about later on. Okay, before getting into the systems biology approaches, I wanted to acknowledge some excellent work that was done by the crystallographers in the team. So during that period of time that we had the MBGC project, more than 16 protein structures were determined. Gary Bushko, I'm happy to see him here. Mike Kennedy and colleagues at EMSL have solved the first structures of the Cyanothece, of a Cyanothece 51142 protein. Tom Smith's group at the Danforth Center in St. Louis solved the structures of 3 ABC transporter solute binding proteins that are crucial for cyanobacterial physiology. I also want to show that they were the ones who started producing or writing papers and getting them published long before those of us who were doing systems biology and modeling. So 2005 we started, and in 2006 already all of these papers started showing up. I'm not going to show you the list of 60-odd papers that we published together, but I wanted to acknowledge the fact that the structural biologists did remarkably good work. What are the structures of? So here is one that Gary's group solved. This is an interesting class of proteins that have repeated 5 residues, and this is the structure. I'm not going to go into the detail of what does what and that kind of thing. It's just a collage of different structures. Here are 2 structures of 2 ABC transporter proteins, one that binds bicarbonate, very important for the growth of cyanobacteria, the other one that binds nitrate, and this is also very important for non-nitrogen fixing cyanobacteria. And so Tom Smith's group solved the structure. Here is the bicarbonate binding protein, and here is the nitrate binding protein. What is important is that Tom's group immediately figured out that the 2 structures look very similar. But there are these important determinants that I did not have time to go after that actually determines that this protein binds bicarbonate, that protein binds nitrate, being in the same space at the same time, okay? And then 2 other elements that are very important in cyanobacterial biology are iron and zinc. And here are the 2 proteins again. They look very similar. They are the ABC transporter proteins. One binds iron with 4 tyrosine and 1 histidine ligand. The other one binds zinc. And that zinc story actually is very, very interesting and unique in biology. And, but I don't have time to go over that. It has 3 histidine residues that coordinate that zinc. So this is the structural part that went in. We wanted to see what the inside of the cell looks like, the cellular ultrastructure. So Michel Liberton and others, and also Lou Sherman's group, were very much involved in electron microscopy analysis of the cellular ultrastructure. So this is a 2 nanometer tomographic slice images of a Cyanothece cell that show the intracellular detail. I checked last month, and this still remains to be the largest tomographic reconstruction of a cyanobacterial cell to date. And this work was published in 2011, but was done during the time that the MBGC project was on. It shows the details, and as you can see that there are lots of membranes inside a cyanobacterial cell. I want to emphasize this because this was, after all, a membrane biology grand challenge project. And so the reconstruction showed in this kind of a picture of what the inside of a cyanobacterial cell looks like. And all I, again, want to show are the thylakoid membranes, where photosynthesis happens. These are the membranes that are all in one contiguous membrane system that wraps around inside the cell, an amazing amount of membrane material that's present in every cyanobacterial cell. Okay, so let's now talk about diurnal rhythm. The Cyanothece diurnal cycle, the way we approach this, and this was very important because you may remember that there were 6 sites, not just here at EMSL, but also these other institutions where we are all going to work on these systems biology projects. So the first thing we decided when we first time met here, probably in this room in 2005, that we all have to have near identical growth facilities. And that gave rise to the idea of having photobioreactors that are identical. One was here, still here, one at Purdue University with Lou Sherman, the other one at our institution at Washington University. These 3 bioreactors were then used to grow the cells under near identical conditions as much as possible. And then every 2 hours, we will sample the culture that's growing, the Cyanothece culture that's growing. The first thing that we noticed, and I'm going to show this kind of a diagram from now on, so let me just spend just a few seconds. This is dark 12 hours, that's light 12 hours, and dark and light. The first thing is that the nice feature about diurnal rhythm or any kind of rhythm is that they repeat themselves. And so this is a very nice feature for physicists, for systems engineers, because you have the same signal coming back, and in this case, every 24 hours. So this takes away the features that are coming from noise, from features that are coming from signal, if we signal average over a period of time. Now, what I show here is that the capacity of photosynthesis is present in light, but in the darkness that goes down. Nitrogen fixation occurs only in the darkness, and then it's not present at all, nitrogen fixation ability, in the light phase, and it repeats itself over and over again. So these were the features that we knew when we started the project. Cyanothece diurnal cycle, photosynthesis and nitrogen fixation are separated temporarily in the same cell. Well, in those days, genome sequencing was a big deal. Nowadays, it can be done in an evening. In those days, it took a long time. And independent assembly, we found certain features, and I'll show you those features in a second, that needed orthogonal approaches to be taken, and that was the use of optical mapping. And we found this, a circle and a linear, two different chromosomes in a cyanobacterial cells to date. As far as I know, this is the only cyanobacterium in which a linear chromosome and a circular chromosome, they coexist. And then there were plasmids in that. We needed the genome sequence for our subsequent experiments with the omics facility. When we did the genome sequencing, we found out that Cyanothece ATCC on the phylogenetic tree is sitting right there. And I just want to point out something again that I'll come back to. This organism that cannot do nitrogen fixation I mentioned to you, Synechocystis 6803, it's very closely related to Cyanothece 51442. You will see why I mentioned that later on. So transcriptomics was the big thing at that time, and at that time it was all based on microarray, and so we did that. And I want to show you the following one. The nitrogenase itself, we could decipher from the genome sequence that there are 35 genes over about a 28 kilobase pair region on the genome that are all related to nitrogen fixation. And when we looked at the levels of the transcripts from each one of these genes, this is the way they looked. I want to draw your attention, this is log ratio. So 7 log ratio drop in the transcript level in light as compared to darkness. Experiments used to be really expensive in those days for microarray analysis, so we repeated that just 2 times. But again, you can see it's a very robust oscillation. Damping is very low. So this is about the transcripts. Later on, Jason McDermott here did a beautiful study of taking those data and converting those to this kind of a model, what we call a read model. And I will describe this to you later on. This is a paper that was published in 2011. This had lots of details of that transcription analysis. Okay, in the paper that we published earlier on in 2008, for analyzing this transcript data, shows this. And what this is that the same read model, each dot is 1 gene, and what happens to it in the dark phase, hour 1, hour 5, and hour 9, and what happens in the light phase, hour 1, hour 5, and hour 9. What is shown here is that, well, red is highly abundant, and blue will be lower abundance. So you can see that in the darkness, energy metabolism, which basically means respiration, it comes up first. And then nitrogen fixation is coming up right after that. This is all at the transcription level. And then here is D9, whereas photosynthesis is very low in the light, in the darkness, and very high in the light phase. So we saw these for the known processes. But the beauty of doing this kind of systems biology analysis is that we can now look at the unknown ones. What are they doing? All the other processes that are taking place in these cells. And when we looked at all of that, did the analysis, we came up with a coarse model of the cell, its metabolic processes in that. So let me walk you through that. Basically what it shows is that take this cell, here's the light, what's happening in the light, here is what's happening in the darkness. And basically the story is very simple. This bug knows how to do a lot of photosynthesis and stores away that fixed carbon in the form of this biobattery, namely glycogen. In the darkness then, the cell is very active, unlike those other not so interesting cyanobacteria like Synechocystis, which just sleeps during the nighttime. This box does a lot of work, and the glycogen is then degraded, respiration takes place, a lot of energy that's produced, and during this period of time, nitrogen fixation occurs and amino acid biosynthesis occurs, and the extra nitrogen is then stored as cyanophycine. So it's two storage bodies, glycogen and cyanophycine. Now comes the Dick Smith and colleagues, their magic, as we heard earlier on. And so up until that time, lots of transcriptional studies that were going on. Our study actually distinguished itself by looking at the protein levels. Now, if you think about the biology, there is no reason why the transcript levels and protein levels or the pattern of changes will follow each other. So we wanted to find out what's going on with the protein levels. And the first one is, again, about the proteins that are involved in nitrogen fixation. Here is the nitrogenase enzyme, and NIF-H, NIF-D, and NIF-K. The H, D, and the K proteins are the important ones. Same kind of pattern. Every 2 hours, you take samples, send them here, and those were then analyzed using the state-of-the-art technique at that time. And basically, we found this. Same kind of read model, same kind of time points, different colors indicate different types of processes. One thing that you will notice right away is that this is much more sparse than what I showed you with the transcript. But this, in my opinion, gave us a much better idea about what metabolic processes are really important inside these cells during this light-dark transition. This is just a diagram that shows, without knowing what they are, the heat map, is that on day 1, if you follow this pattern, you will see on day 2, largely the same pattern is emerging. That gave us confidence that what we are seeing at the protein level is really, a cyclic phenomena that's happening. And when we looked at those proteins, we could easily figure out which metabolic process inside the cell is getting controlled by this diurnal process. And I'm not going to go into the details of that. Here is one, about 19% of the proteins that we detected are involved in photosynthesis and respiration. About 25% belong to central intermediary metabolic pathways. These were the conclusions that we came to from those studies. Coming back to nitrogenase, here is what we saw with the transcripts, and I show only a few of them here now. And here is what we saw with the protein. This is still really intriguing in biology, because this is one of those processes where the protein levels are following in tandem way the levels of the transcripts. So this was a nice way to think about the biology during nitrogen fixation in this organism. So Cyanothece diurnal cycle, we can summarize it as photosynthesis results in oxygen production during the day. High rates of respiration generate low intracellular oxygen tension during early night hours. And oxygen-sensitive enzymes such as nitrogenase can then function in the same cell. And this repeats itself every day. And so summarizing then, first thing that to remember then, from the point of view of biomass production and everything, Cyanothece can store and later mobilize fixed carbon. This has enormous implications for production using cyanobacterial kind of system. And the other one is this business. Cyanothece cells alternate between high and low intracellular oxygen tensions. So all the energy requiring processes, not just nitrogenase function, but also hydrogenase and other things that are very near and dear to our hearts, they should also, in this kind of environment, flourish. So this is something that we figured out. So when I think about the MBGC project, those 5 years of a lot of fun, a lot of experiments doing together, I see that as the result is this. It resulted in a comprehensive systems level and understanding of the lifestyle and metabolic capacities of an oxygenic photosynthetic bacterium under diurnal light-dark conditions, mimicking the natural environment of such autotrophic microbes. This was the first time we had a comprehensive picture of that. Okay, so there are lots of other things that I could have discussed. as I said, there were lots of papers that were published at that time, many more that are really noteworthy, but I use just these examples. And let me spend a minute or two on what's happening post-MBGC. So as I mentioned to you, I came to EMSL and basically got hooked to this environment. And ever since then, all of us together have been conducting experiments together through different kinds of user proposals, and I shouldn't say that, even without user proposals. And we keep on publishing. And so you will see that this is just one area, global dynamic proteomics. Ariel et al. 2011, I picked this one because that's just past the 2010 MBGC, 2012. And I was going to list all of those and I said, no, I better stop. So that's another set of past those 50 papers, lots of papers, including the latest one about 2 months ago. And it continues. One of the things that I was looking forward to is to meeting with John Jacobs and just talking about, okay, so the data for the next paper, how is that going? So we are on a roll and we are not stopping. That's the bottom line. Lot has happening with Cyanothece. We now have CRISPR-Cas working with CRISPR-CPF1 working for Cyanothece, and we can make mutants now in a very directed way. And so this is going to open up other opportunities, and these materials can then be interrogated by using the advanced facilities here at EMSL. There is this kind of synthetic biology project that we are getting involved in, where we are creating or designing nitrogen fixing ability in oxygenic photosynthetic cells. And the simple thing is, let me just go to the bottom line, is instead of doing this in plants, which is the ultimate goal, we just said, well, how can we make this boring organism Synechocystis 6803 fix nitrogen? So borrow the parts from Cyanothece, and with the modern synthetic biology tools, we could put all of those clusters in there, and Synechocystis 6803 has become now nitrogen fixing. So this is the kind of thing that's happening now in this area. And the design principles of these can then be used to make crop plants that fix nitrogen. I want to end with this kind of a scenario in the future. Why do all these things? And one of the things is always attractive is that if we can use photoautotrophic systems as production systems for all the good things that we can make. Nowadays, we do that in a two-step manner. The photosynthetic systems make sugar or carbohydrates. Those are then used by E. coli or yeast, and then we make all the biologically make all the products. Why can't we make it in one step? And a scenario in the future addresses that. So the available land, we are sitting in that time later in this millennium. Available land now must provide not only food, but also the biofuels and other petrochemicals that were once cheaply available from underground. This is the scenario. One solution is engineered cyanobacteria, which can grow on marginal lands and use brackish or ocean water unsuitable for most conventional crops, possibly reclaiming land recently lost to desertification. These are good things. And they can do so at a photosynthetic efficiency, this we know, far higher than corn, switchgrass, and other favorite crops. This part is already known. And the last thing is creative food technology might make cyanobacteria-derived cuisine taste the same as chicken or beef, or even better, with consequent 15-fold reductions of energy use relative to animals. These are not my quote, my writings. George Church wrote it in this book called Regenesis. So what would EMSL do in the future? These are my humble opinions. High-end instrumentation that are well integrated with the state-of-the-art computational infrastructure. This is absolutely essential. Probing behaviors of single cells, advanced imaging of tiny microbes. I'm sorry that Sunny Shi could not attend. I really wanted to hear his views. This is important, rapid analysis of biological consortia in their near natural environments. That's important. And then bio-inspired fabrication. No reason why synthetic biology cannot be coupled with synthetic chemistry. And again, to probe those materials, EMSL is well positioned. And finally, I will say this: resources targeted towards engaging scientific teams with diverse expertise to probe cutting-edge DOE-relevant biological questions. I want to end with this and just take a second to say the following. This is from 2007, and those who are on this picture and present here, compare yourself now and then. I wanted to do, guess who kind of thing on this, but I'm deeply indebted to the MBGC project and EMSL in allowing me to be a part of the scientific community. I think that's the most important thing in systems biology. It's not the analysis or the data, it's interaction between different scientists. So we started with, as I mentioned, not knowing who Dave Copinol is, that just as an example. And then I arrived here yesterday and Dave says, the wineries are there. And there is no reason for us to discuss anything at that level. And when we are there, we are discussing what is the next big experiment. And that thing is continuing through all these individuals and continues to percolate. And I will sincerely hope that EMSL, not as its 100% of its efforts, but at least some part of its effort goes towards, again, allowing maybe another team with diverse expertise to come and take advantage of the fantastic toys that are here. Thanks for giving me the opportunity.